Hi, my name is Claire Klein, and today I will be presenting on the structurally specific lexical diversity of minimally verbal children with autism. This is work that I led when I was working at Dr. Sophie Kim's lab at the Center for Autism and the Developing Brain at Weill, Cornell. This work was funded by the National Institute of Mental Health. Structurally specific lexical diversity comes from a sentence-focused framework. This approach measures the variety of words used in structurally defined positions of sentences, like subjects and verbs. In research with typically developing children who are slow to begin speaking, structurally specific lexical diversity has been found to be a clinically meaningful and developmentally sensitive measure of language development. To measure structurally specific lexical diversity in children, we can use the number of unique subject-verb combinations. As children learn basic clause structure, they discover that words are grouped into different parts, like subject and verb phrases. Then we start to see these subject-verb combinations appear in childlike sentences, which then develops further into adult sentences. A child may start out by saying, want more, and then add in a subject, mouse want more, and then add in grammatical markings, the mouse wants more cookies. To measure these changes, we can measure the number of unique combinations of a lexical verb and a pronoun or noun in the subject position. Here, mouse want and the mouse eats would both be considered unique because the child is using the same subject but with different verbs. In non-clinical samples of children who were late to begin speaking, parent input, specifically toy talk, was found to promote child lexical diversity. Toy talk is a strategy to increase the variety of unique subject-verb combinations used by parents. Toy talk consists of giving the toy its name and talking about the toys. Together, toy talk strategies promote the use of lower frequency nouns, such as names of specific toys, compared to using pronouns that are higher frequency. For example, instead of using the pronoun in he needs to eat, parents are encouraged to say, the bear needs to eat. This helps children differentiate these different parts of speech better. Parent input, specifically varied use of unique subject-verb combinations through toy talk, has been found to support vocabulary development for children with typical language development. This suggests that interventions for ASD can focus on ensuring rich parent input, which may increase children's lexical diversity. Because impairment in social communication is a core feature of autism, many young children need support in their early language skills. Although expressive language is a common focus of parent-mediated interventions, a more targeted treatment model, like toy talk, has not been validated in children with ASD. Therefore, this study explored the use of toy talk in parents within naturalistic developmental behavioral intervention for autism. Therefore, our study had two aims. First, we aimed to examine changes in lexical diversity in children with autism who were receiving parent-mediated interventions. Second, we aimed to examine predictors of lexical diversity changes in these children with autism during intervention. Participants included 50 toddlers and their caregivers, and pairs of videos were coded 6 months apart during treatment. All participants were receiving parent-mediated NDBIs, naturalistic developmental behavioral interventions. The coders who rated language features were masked at time points in treatments and transcribed the videos using SALT. Coders tallied the number of unique subject-verb combinations in children and parent use of toy talk. So our first objective was to examine lexical diversity changes in children. Based on results of our mixed model analysis that you can see here, children as a group showed significant improvements in lexical diversity over the course of 6 months of behavioral intervention. We also see that other covariates, baseline parent toy talk, nonverbal IQ, and age, are predictive of lexical diversity across both time points. Specifically, higher parent use of toy talk, higher nonverbal IQ, and older age predicted better lexical diversity. However, when we look at the individual level data, we see tremendous heterogeneity in the magnitude of improvement in subject-verb combinations across individual children. This figure shows the variability and changes in lexical diversity, with each participant on the y-axis and individual change on the x-axis. The darker dots are subject-verb combinations at baseline, with the bar and lighter dots showing subject-verb combinations 6 months later. For children who never develop subject-verb combinations, their dots are all lighter. Given the heterogeneity, we are particularly interested in those children who started with no subject-verb combinations. We found that the number of subject-verb combinations in some children remained stable at 0, while subject-verb combinations in others improved over time. Next, we looked at the clinical and behavioral features that differentiated these two groups. Here, we see children who show an improving trajectory for subject-verb combinations have higher nonverbal IQ, higher verbal IQ, and lower symptom severity at baseline. We also see that those children who show improving subject-verb combination and trajectories are more likely to have parents who use higher toy talk at baseline, and there were no differences in baseline child age between the groups. Not surprisingly, higher baseline mean length utterance measured in words or morphemes, higher number of different words, and higher number of total words at baseline were also predictive of those that went on to develop subject-verb combinations. In summary, our results indicate that children with ASD receiving parent-mediated intervention show improvements in structurally specific lexical diversity as a group. But there is a high level of individual variability in the magnitudes of these changes. We also found that outside of the impact of parent language input, higher cognitive skills and lower autism symptom severity may facilitate early trajectories of grammatical growth in young children with ASD. Based on these results, an initial intervention study of toy talk for children with ASD is ongoing at Dr. Kim's lab. And we would like to thank the families who contributed their time and efforts to this research. And thank you to Dr. Sophie Kim for providing me with the opportunity to lead this project, and to the rest of the ASD and Developmental Science Lab at the Center for Autism and the Developing Brain.
Good afternoon. Today, I will discuss delay of gratification and inhibition in children with Williams syndrome, which is one component of my dissertation. We would like to thank the members of the Neurodevelopmental Sciences Lab and the families who have participated in our research. Our research is supported by the Williams Syndrome Association and the National Institutes of Health. During my presentation, I will give a brief introduction of Williams syndrome, as well as hot and cool inhibition. Then I will describe the results of the current study where we evaluated children's inhibitory control using a gift delay of gratification task. Williams syndrome is caused by a microdeletion of 26 to 28 genes on chromosome 7. It is characterized by specific cognitive and behavioral profiles. Children with Williams syndrome often exhibit mild to moderate intellectual disability and have impairments in behavior and emotion regulation. They often have deficits in inhibitory control, which is the ability to overcome an automatic or dominant response. Within inhibitory control, there are two separable yet interrelated hot and cool subdomains. Hot or effective inhibition is active in motivationally and emotionally significant circumstances, such as resisting temptations. Cool or cognitive inhibition is active in emotionally neutral circumstances, such as focusing attention. Historically, inhibitory control research has focused primarily on cool inhibition. This trend is also observed in research that includes participants who have Williams syndrome. Several studies have focused on cool performance-based measures. These studies have indicated deficits in inhibition during emotionally neutral contexts. Research on hot inhibition has predominantly used delay tasks. In these tasks, an enticing reward is presented, and the child is to delay gratification by inhibiting a behavior while waiting on the examiner. In typically developing children, the ability to wait is facilitated by emotion regulation. In children and young adults with Williams syndrome, emotion regulation has been shown to be positively associated with practical problem solving. However, to my knowledge, there's no published study that has evaluated inhibition using a delay task for individuals with Williams syndrome. In the current study, we directly measured hot inhibitory control using a gift delay task. Relations between inhibition, intellectual ability, and parent ratings of behavior and emotion regulation were evaluated. Two aspects of emotion regulation, shifting and emotional control, were also explored. Participants included 33 6 to 8-year-olds. All children had genetically confirmed classic link deletions of the Williams syndrome region. We used the DAS2 GCA, or General Conceptual Ability Standard Score, to evaluate overall intellectual abilities. We used the Brief-2, which is a parent report measure of executive function. The current study focused on the emotion regulation and behavior regulation indices. Higher T-scores indicate greater difficulty. Mean GCA was consistent with previous reports of intellectual abilities for children with Williams syndrome. For emotion and behavior regulation, mean T-scores were elevated. Standard deviations were similar to the general population, and scores ranged from no impairment to severe impairment. Here, you can see screenshots of a participant at the beginning of the gift delay task and as the task progressed. Before the task, the examiner told the child she had a gift, but she had forgotten to wrap it and wanted it to be a big surprise. So the child was instructed to remain seated and not look or peek. Then the examiner went out of the child's view to loudly wrap the gift for 60 seconds. The task goal was to delay gratification by inhibiting the urge to peek at the examiner or the gift while it was being wrapped. The task was filmed and behavioral coding was conducted. Categorical pass-fail ratings were assigned. If the child delayed gratification and looked away from the gift while it was being wrapped, the child passed. If the child peeked, the child failed the task. We explored inhibition as indicated by overcoming the urge to peek at the examiner or the gift while it was being wrapped. The circles indicate when the child was successfully inhibiting and looking away from the gift. To characterize inhibition, we coded and then summed the amount of time that children looked away from the gift. This allows us to capture variability in inhibition. A higher time indicated greater inhibition. Overall, 33% of the children passed the task, meaning they successfully delayed gratification and looked away from the gift for the entire 60 seconds. For the 22 children who failed, they were able to inhibit and looked away from the gift for a mean total of 31 seconds. However, there was a wide range in the looking times. We conducted a multiple regression predicting inhibition as measured by the total time children looked away from the gift. Intellectual abilities and emotion regulation were significant predictors. No significant effect was found for behavior regulation. After controlling for behavior regulation, higher intellectual ability and better emotion regulation predicted inhibition. To explore which components of emotion regulation were most related to task performance, the two subscales of the Emotion Regulation Index were evaluated. Shift evaluates the ability to move smoothly from one facet of a problem, task, or situation to another as the circumstances demand. Emotional control evaluates the ability to regulate emotional responses. For the current sample, mean T-scores for both skills were elevated. Standard deviations were similar to the general population, and scores range from no impairment to severe difficulties in shift and emotional control. We conducted a multiple regression predicting inhibition as measured by the total time children looked away from the gift. Intellectual ability and shift were significant predictors. No significant effect was found for emotional control. After controlling for emotional control, results indicated higher intellectual ability and better shifting skills predicted inhibition. In sum, deficits in inhibitory control were evident in 6 to 8-year-olds with Williams syndrome. Only 1/3 of the participants were able to successfully delay gratification and looked away from the gift for the 60 seconds it was being wrapped. When evaluated concurrently, only parent report of emotion regulation and not behavior regulation was significantly related to task performance. Consistent with research on typically developing children, hot inhibition was facilitated by emotion regulation. This supports that the gift delay task is a measure of hot inhibitory control for children with Williams syndrome. Within emotion regulation, better shifting ability was significantly related to better hot inhibitory control. Emotional control was not related. This indicates the ability to shift or flexibly problem solve facilitates effective regulation strategies, which makes it easier for the child to exercise self-control and inhibit impulses. Thus, for children with Williams syndrome who have impairments in shifting, get stuck focusing on the temptation or the gift. This makes it harder to overcome impulses and is reflected in the deficits in hot inhibitory control observed in children with Williams syndrome. This brings me to my future directions. Inhibitory control underlies many aspects of daily life, such as adaptive function, social skills, school achievement, attention, and persistence. Children with Williams syndrome often have impairments in these facets. Research in typically developing children have identified differential relations between hot and cool inhibition within outcome measures. In my dissertation, I'm exploring if deficits in hot inhibition and cool inhibition, or both, may be related to some of the difficulties observed in early school-age children with Williams syndrome. Thank you so much for listening.
Hello, today I'm presenting some of our research looking at different ways of measuring sensory processing in autism. Of course, sensory processing in autism is very commonly atypical, and this is impactful on quality of life and other variables. But there's a lot of different ways that one can measure sensory processing in autism, and we may need more research using multiple different kinds of measures, seeing how they relate to one another. So the present study is using 3 different kinds of measure: event-related potentials, parent and self-report questionnaires, as well as psychophysical thresholds in the auditory and tactile modalities in adolescents aged 11 to 14. We have 46 autistic and 21 typically developing participants. There is a group difference here in cognitive ability, please note. And then our questionnaire measures include the sensory experiences questionnaire and the sensory profile, which are both parent report questionnaires, as well as the adolescent adult sensory profile, which is a self-report measure. And then we have auditory detection thresholds to pure tones of 125 through 8,000 hertz. And also tactile static stimulus detection thresholds. So we applied von Frey monofilaments to the right index finger in ascending and descending order based on filament thickness. And these are only being reported from the right-handed participants. We also have tactile spatial resolution thresholds. So here, gratings of bars and grooves were applied to the right index finger. They're of varying groove thickness, and they were applied in different orientations, so 90 degrees and 0 degrees essentially, and participants were asked to report the orientation. They were presented with 10 trials at each grading width until the threshold was identified. And again, these are only being reported from right-handed participants. Finally, we have event-related potentials. So These are auditory noise bursts and somatosensory taps. The somatosensory taps are delivered to the index finger here, which is also doubling as a response button because this is a speeded reaction time task. People are responding to all events as quickly as they can. And there's 130 stimuli per condition intermixed, and other stimuli being presented here include visual and multisensory stimuli because this is part of a multisensory integration task. So in terms of our results, we did find robust group differences on all of the questionnaire subscales, with the exception of the adolescent adult sensory profile sensation seeking subscale. And we don't think this is anything to do with this being a self-report scale. We think it has something more to do with the items, because the items don't really seem to tap into necessarily the sorts of things that provoke sensory interest in autistic people, whereas items that seem more closely relevant to that, like those on the sensory interest scale of the sensory experiences questionnaire, are showing robust group differences. So this is probably reflecting that the sensory profile was not primarily designed for use in autism, whereas measures like the sensory experiences questionnaire were. We did not see, nearly such robust group differences in hearing thresholds. When we use permutation testing based on, finding the maximum statistic across all of these different frequencies in both ears, and then comparing that effect to a permutation distribution of effects that would be expected by chance, we did see a non-significant trend towards a group difference, but that was primarily driven by the left ear, where you can see perhaps slightly lower thresholds in autism, and the left ear was always tested first. So probably this is just reflecting elevated attention early on, not so much perhaps an actual difference in sensory acuity per se. And again, it wasn't significant. And there was nothing going on in terms of group differences in tactile thresholds. We did see that auditory TB, so that's also called the N1C component, its amplitudes were larger and latencies were faster in typically developing participants, although those effects were not significant after covariating for cognitive ability. We also saw that amplitudes of the somatosensory P60 were larger in typically developing participants, but again, not quite significant after covariating for cognitive ability, and there's no difference in latency here. Now we want to look, of course, at the associations between these measures, specifically in the autism group. And for the questionnaires and the psychophysics, we focus specifically on low threshold enhanced perception scores that might be related to behavioral thresholds, we thought, and in the specific modality in question, so auditory or tactile, but yet we saw no correlations with any of these questionnaires. Similarly, when we looked again in the autism group at correlations between the event-related potentials and the questionnaires, focusing on the overall scores in the auditory or tactile modalities, whichever the case may be, we did not see any effects. However, we saw an effect that we were not expecting at all, that participants with a larger or more negative TB response actually ended up having higher thresholds in the autism group. And that was a pretty robust effect, not driven by these outliers here, I should say. And we were a bit puzzled as to how to explain it, but one possibility is that this is reflecting the reaction time task, that participants are trying to respond as quickly as possible. If you have a low threshold, maybe you're able to initiate that response earlier and you don't keep processing the stimulus quite as much, whereas somebody who has a higher threshold maybe needs to keep that neural response going a bit longer. Unfortunately, we can't look at some of the earlier event-related potentials very well here because a lot of them are recorded at the vertex, and there's a developmental shift such that some participants have positivities and others negativities over the same electrode sites at the same time. Another interpretation of this effect, though, is that it reflects compensatory processing. There is prior research in individuals who are hard of hearing that shows enlarged neural responses to sounds. It's unclear whether you would necessarily see similar compensatory processing in individuals with normal hearing thresholds, but it's certainly one interpretation. We didn't see any associations with somatosensory ERPs on the tactile thresholds. So to summarize, it certainly seems as though the questionnaires are showing the most robust group differences and perhaps most relevant to the real world sensory experiences of autistic people. The other measures didn't really seem to be converging with the questionnaires. Although it is possible that some questionnaires are not as suited for use in autism than others, that measures that have items that were designed in autism contexts might be better for this population. We also saw this, again, very unexpected association between auditory TB amplitudes and hearing thresholds. So we think further research probably be helpful to figure out what's going on here, but it could be a very important factor for auditory ERP studies in autism because this could point to a confound. Hearing thresholds could be a confound here in ERP research. A couple of other future directions. Here we didn't examine discomfort towards stimuli in lab, just detection and discrimination. So that could be interesting, might be more relevant to real world sensory experiences and might converge better with questionnaires. And it would also be interesting to examine questionnaires that drill down more deeply into specific aspects of processing here, because here we were just looking at questionnaires that look at things like generalized hyper and hyposensitivity, but pointed out in a recent review by Zach Williams, you can get a lot more specific with that. So to conclude, just of course thanking everybody who made this research possible, and also thank you for your attention, and hope you found this talk interesting.
Today, I'm going to talk about a study we did examining verbal ability as a potential moderator between mother-child relationship quality and child maladaptive behaviors in ASD. To recognize our funding, this study was supported by a core grant to the Waisman Center, as well as an R01 to Dr. Marcia Malik and an F31 to myself. So we know that there's a well-established relationship between child maladaptive behaviors and mother-child relationship quality in ASD, and that this relationship is bidirectional in nature. But the relationship between child maladaptive behaviors and child verbal ability is less clear and has focused on toddlers and young children on the spectrum. And similarly, little is known regarding whether mother-child relationship quality is impacted by child verbal status. So does the child's ability to speak impact the relationship quality between the mother and the child? And further, it's not clear whether the relationship between child maladaptive behaviors and mother-child relationship quality is moderated by child verbal status. And this is particularly important since we know that approximately 30% of autistic individuals remain minimally verbal into adulthood, and this percentage grows among those that have co-occurring intellectual disability. So for this study, we asked 3 questions. First, does the relationship quality between mothers and their adolescent and adult children differ based on child verbal ability? Second, do child maladaptive behaviors differ from minimally verbal versus verbal individuals? And third, does verbal status moderate the association between mother-child relationship quality and child maladaptive behaviors? This study included 219 autistic adolescents and adults with ID from a larger longitudinal study, and the participants were divided into 2 groups based on verbal status using 1 item from the Autism Diagnostic Interview revised. And so with this item, the examiner asks the mother a number of questions and probes to get at the current language and speaking abilities of the individual on the autism spectrum. And this item is scored as a 0, 1, or 2, where 0 equals daily functional use of phrases. And we categorize this group as the verbal group. And a score of 1, which is no functional use of phrases but uses speech daily, or 2, less than 5 words total or speech not used daily. And so individuals who scored a 1 or 2 were classified as minimally verbal. And classifying verbal status in this way is consistent with previous research. So in doing this, we ended up with 143 verbal autistic adolescents and adults and 76 minimally verbal autistic adolescents and adults. And the groups were largely similar on demographic variables, apart from maternal education, where we saw higher levels of maternal education among the mothers of the verbal compared to minimally verbal adolescents and adults. And so to contextualize this a bit, Mothers of verbal adolescents and adults on average had between an associate's and a bachelor's degree, whereas mothers of minimally verbal adolescents and adults had between some college with no associate's degree and an associate's degree. To measure maladaptive behaviors, we use the problem behavior subscale of the SIBR, and this probes 8 specific behaviors. So when we compared across groups, we looked at the presence or absence of these 8 behaviors. And then we also looked at the general maladaptive behavior score, which takes into account the presence or absence of these 8 behaviors, as well as the frequency and severity of these behaviors as well. And then we measured mother-child relationship quality through 4 indices. We looked at 2 positive aspects and 2 negative aspects. For positive aspects, we looked at mother-child closeness using the Bengtson Positive Affect Index. And this is a 10-item parent report scale that looks at different aspects of closeness, such as affection and understanding. We also used the 5-minute speech sample developed by Magana and colleagues, which uses a standardized prompt to ask the mother to speak about her child for 5 minutes without interruption. And then using these recordings, the number of maternal positive remarks was identified. And then in terms of negative aspects, we used the burden interview to measure maternal burden. And this is a 29-item parent report scale, which assesses subjective burden associated with caregiving demands. And then we also used the 5-minute speech sample to code the total number of maternal critical remarks regarding her child. For research question 1, we found no group differences in mother-child relationship quality across the 4 variables that we probed. And for research question 2, we found 2 group differences. So on the x-axis, you can see the different problem behaviors, and on the y-axis, you see the percent of mothers who endorse these behaviors in their adolescent or adult child. And the gray represents the verbal group, and the blue represents the minimally verbal group. So we see that mothers were more likely to endorse disruptive behaviors and socially offensive behaviors when the child was verbal versus minimally verbal. And then also there was not a difference between groups on the overall general maladaptive behavior score. When testing for moderation, we found that verbal status did not moderate the association between child maladaptive behaviors in positive aspects of mother-child relationship quality, so mother-child closeness and maternal positive remarks. But we found that verbal status did moderate the association between child maladaptive behaviors in negative aspects of mother-child relationship quality. So I'm going to dive into these negative aspects, but I'm happy to share the tables with anyone if they're interested in the positive aspects as well. So what you see here on the x-axis is child maladaptive behavior score, and the y-axis is predicted maternal burden. The blue line represents the minimally verbal group, and the gray line represents the verbal group. And then the dashed line represents the cutoff for clinical level of maladaptive behaviors. And so what we found was that there was a stronger association between maladaptive behaviors and maternal burden in the verbal group as compared to the minimally verbal group. When predicting maternal critical remarks, we found that at low levels of maladaptive behaviors, that mothers produced a similar number of maternal critical remarks. But at high levels of child maladaptive behaviors, mothers of verbal adolescents and adults produced a greater number of critical remarks compared to mothers of minimally verbal adolescents and adults. And so in conclusion, child verbal status does not impair mother-child relationship quality among those with ID. And this is particularly important because there is a large subset of autistic individuals who are minimally verbal and have ID. And this really speaks to the resiliency of the mother-child relationship among these dyads. And then we found that verbal status moderated the association between child behavior problems and negative aspects of mother-child relationship quality, but not positive aspects. And perhaps this speaks to the fact that the interpersonal disruption arises not from level of impairment, so not from speaking ability, but rather from the presence of maladaptive behaviors in the child. So perhaps it's the fact that the presence of disruptive and socially offensive behaviors really strongly impacts mother-child relationship quality, although this needs to be probed further. we also need future work that uses longitudinal studies to look at these variables and includes autistic individuals both with and without ID. So lastly, I'd like to acknowledge the families as well as the study examiners and interviewers who were all part of this research. And I'm happy to share tables as well as references with anyone who's interested. Thanks.
Hello, I am so excited to be a part of this symposium today and thrilled to share with you some of the work coming from our lab at Loma Linda University, part of which stems from my dissertation project. So research indicates that parents of children with developmental delays experience significant stress, and this is more than parents of typically developing children. When we look more closely at specific populations, we see that parents of children with autism typically report the highest levels of stress. Unsurprisingly, there are a number of negative consequences of stress, which you can see here, all of which to say that stress has a serious impact on parents themselves. But one thing to consider is that parent stress also impacts child outcomes. So parents that are stressed have report that their children have increased social and behavioral difficulties. And this seems to have a reciprocal relationship with parent stress. We also see that parents are more controlling, intrusive, and assertive when they're stressed, which can ultimately impact their development of self-regulation and social skills, and may also contribute to problem behaviors, which we know and we just discussed can contribute to parent stress. One thing we do know is that for managing stress, a spouse or romantic partner may be a source of emotional support. However, we know very little about the relationships between parents of children with autism. What we find is that parents of children with ASD report lower marital satisfaction and higher risk of divorce than parents of typically developing children. However, we also see that relationship quality is associated with a number of positive outcomes, including increased warmth and responsiveness, reduced parent-child conflict, higher quality parent-child dyadic interactions, and decreased parenting stress. So all that to say that it's worth understanding how we can improve parents' relationship quality to help them better manage their stress. The spillover hypothesis is the idea, kind of what we're hinting at, that the day-to-day conflict in parents' marital interactions may spill over into their interactions with their child. So essentially, tension or particular behaviors that parents exhibit together in their relationship, in their interactions together, may ultimately spill over into parents' interactions with their children. And this might occur within a single day or over time. For parents of children with autism who we already know to be experiencing higher levels of stress, this effect may be exacerbated. We do see evidence of spillover in the general population with a positive relationship between marital quality and adaptive parenting behaviors and parenting experiences. We also see that parents of typically developing children are more likely to experience strained parent-child interactions following a day with more marital tension. We have limited evidence of spillover among parents of children with autism. However, Dr. Hartley has demonstrated that a negative marital day was associated with a more stressful parenting day. And specifically, mothers experience fewer positive interactions with their spouse on days when they experience more parenting stress. So the current study aims to further understand these relationships by utilizing a daily diary survey to examine the relationship between parenting and marital stress among parents of children with autism. This nested data structure allows us to explore more in depth these relationships at the between and within parent levels. Our research aims to examine the time-lag spillover effects of marital and parenting stress at the between and within person levels. Our first aim is to determine if parenting stress predicts the quality of next day marital interactions. And the second aim is the opposite. Does quality of marital interactions predict next day parenting stress? So for this study, we use data from the Stress Reduction Techniques for Enhancing Parenting Skills, or STEPS program at Loma Linda. It is a research study designed to examine the effectiveness of mindfulness-based stress reduction as compared to psychoeducational support for reducing stress for parents of children with autism and decreasing child behavior problems. Essentially, the children had to be between 3 to 5 years old with an autism diagnosis. Then parents were randomized to group intervention and completed assessments at baseline, post-treatment, and 6 and 12-month time points. For the daily diary portion of the study, the parent that was identified as the primary caregiver was eligible to participate if they were in a current relationship and living with their spouse or partner. And that could be dating, married, or separated. And this was elective, so parents gave consent and agreed to participate in this above their regular participation in the study. Parents were then sent a link each day of their survey completion, a link for the Qualtrics survey, and were asked to complete the diary questions once per day for 14 consecutive days and at the same time each day. Just to review this overall study design, so parents completed a baseline assessment, were randomized to MBSR or psychoeducation groups, then completed post-treatment 6 and 12-month follow-ups, and the daily diaries were completed after randomization and after the post-treatment assessment. The questions that are relevant for analyses today are the quality, two components of the daily diary, which is measuring the quality of marital interactions, specifically the occurrence of 8 positive and 8 negative interactions with their spouse or partner each day. And then an item measuring parenting stress, which was a single item on a 7-point Likert scale, assessing overall how stressful were your parenting experiences with your child each day. For the current analyses, we ran 3 timeline multi-level models with using full maximum likelihood estimation. In each model, we controlled for the autoregressive effect of the previous day measurement of the outcome variable. The predictor of interest was partitioned into its between and within person components. And our level 1 predictors were person-centered, and level 2 continuous variables were grand mean centered. Here's an overview of our demographics. Essentially, our parents were about 35 years old, mostly biological mothers, Hispanic or Latino, married, and with at least some partial college education. For our first aim, on average, previous day negative marital interactions significantly and positively predicted next day negative marital interactions. On average, parents reporting higher levels of parenting stress across days tended to report more negative marital interactions. And within parent fluctuations in previous day parenting stress did not significantly predict differences in next day number of negative marital interactions. And then for the next model with parenting stress predicting positive marital interactions, on average, previous day number of positive marital interactions significantly and positively predicted the next day number of positive marital interactions. However, parenting stress did not significantly predict differences in number of positive marital interactions within or between parents. For our second analysis, on average, parenting stress did not significantly predict next day parenting stress. Parents who reported more negative interactions across days tended to report significantly higher levels of stress. And then within parent fluctuations in previous day negative marital interactions did not significantly predict differences in next day levels of parenting stress. And finally, for positive interactions predicting parenting stress, neither previous day between or within person differences in positive marital interactions significantly predicted differences in next day parenting stress. So putting all of that together, essentially previous day negative or positive marital interactions predicted more of the same. Parenting stress and negative marital interactions flowed bidirectionally. Parenting stress was not associated with positive marital interactions, and parenting stress did not fluctuate with day-to-day changes in marital interactions. So pretty consistent there. So overall quality of parents' marital interactions was predictive of their parenting stress, particularly negative interactions, and parenting stress seemed to flow bidirectionally. Having a more positive marital interactions was not predictive of a reduced parenting stress, so parents may need more targeted support for managing stress. An intervention designed to help parents better manage their stress may help improve relationship quality as well. So for our next steps, we do plan to rerun this with our full sample now that we have recruited all of our participants for the full study. We will also then examine post-treatment differences in spillover to determine if there are any treatment effects. And then we'll examine some possible cultural influences given the demographic of our sample. So just a quick shout out to everybody mentioned here who this work could not be possible without. And with that, I will happily take any questions. Thank you.